Hello everyone, your Gen Z teacher is here to tell you some important MCQs of biotechnology principle and processes. So here our first question. Which of the following is not a principle of biotechnology? Option A, the use of living organisms or their products to create or modify products or processes for specific use. Option B, the understanding and manipulation of DNA and genes. Option C is the use of microorganisms to produce useful products such as antibiotics and vaccines. The use of chemicals to synthesize biological molecules. So options A is very much correct because we know that the use of living organism or their product to create or modify product or process for specific use, it is already given in NCERT as well, and the B is also right. We manipulate and understand the genes and DNA. The C is use of microorganism to produce useful products जैसे कि vaccines, antibiotics. So these three options are very much correct. The option D is not correct. It is stating the use of chemicals to produce biological product, which is not correct. The next question is which of the following is not a tool of recombinant DNA technology? Options are restriction enzymes, plasmids, ligases, PCR machines. So the first option is very much correct. Restriction enzymes cut the DNA at specific points, and plasmid is also a vector. Ligases joins the cut part of DNA, and the D, which is PCR, which is used for DNA amplification, not for recombinant DNA, right? So the correct answer for this question will be PCR. PCR is not a tool of recombinant DNA technology. Yeah, D is correct. The next question we see is which of the following steps is not involved in gene cloning? First is isolating the desired gene from the source organism. B. Inserting the gene into a vector. C. Transforming the vector in a host cell. Selecting for transformed cells. तो पहला जो ऑप्शन है वो आइसोलेशन ऑफ डिज़ायर जीन जो हम करते हैं रिकॉम्बिनेंट डीएनए बनाने के टाइम, उसके बाद उस जीन को एक वेक्टर में हम ट्रांसफ़र करते हैं, इंसर्ट करते हैं, फिर उस वेक्टर से होस्ट तक ट्रांसफॉर्मिंग होती है, ट्रांसफॉर्मेशन जिस प्रोसेस को कहते हैं. सो पहले तीन ऑप्शंस बिल्कुल करेक्ट हैं. द डी ऑप्शन विच इज़ सेलेक्टिंग फॉर ट्रांसफॉर्म्ड सेल्स, ये ऐसा कुछ नहीं होता है. सो द डी ऑप्शन इज़ नॉट करेक्ट. सो वी विल गो विद द आंसर डी. Let's check it out. So correct option is D. The next question is which of the following is not an application of biotechnology? So the options are production of genetically modified crops, development of new drugs and vaccines, bioremediation of pollution, or production of renewable energy. So the first option जो कि है production of genetically modified crops, which is very much true, and B is development of new drugs and vaccines. This is also correct. Bioremediation of pollution. Option B, development of new drugs and vaccines, correct. Bioremediation of pollution, correct. And option D is not correct. So the right option for this question is option D. So yeah, D is the right answer for this question. The next question is which of the following is the correct order of steps in southern hybridization technique? So options are gel electrophoresis, hybridization, southern blot transfer, hybridization, gel electrophoresis, or hybridization, southern blot transfer, or gel electrophoresis. Or option D, gel electrophoresis, then sudden blot, then transfer, then hybridization. जो करेक्ट सीक्वेंस है फॉर द होल प्रोसेस ऑफ हाइब्रिडाइजेशन टेक्निक, सदर्न हाइब्रिडाइजेशन टेक्निक का, दैट इज़ ऑप्शन सी. फर्स्ट वी डू हाइब्रिडाइजेशन, देन उसके बाद सदर्न ब्लॉट पे ट्रांसफ़र, देन जेल इलेक्ट्रोफोरेसिस. सो द ऑप्शन फॉर दिस क्वेश्चन इज सी इज़ करेक्ट आंसर फॉर दिस क्वेश्चन. C is the correct answer for this question. Let's see some other question. Which of the following is not a type of restriction enzyme? A. Type 1, type 2, type 3, or type 4? So correct answer for this question is type 4, which is D. टाइप 1 रिस्ट्रिक्शन एंजाइम भी होती हैं, टाइप 2 भी होती हैं, टाइप 3 भी होती हैं, पर टाइप 4 रिस्ट्रिक्शन एंजाइम नहीं होती हैं. सो करेक्ट आंसर फॉर दिस क्वेश्चन इज ऑप्शन डी, which is type 4. Let's move on to next question. Which of the following is not a function of a plasmid? Replication, gene transfer, gene expression, antibiotic resistance. So the correct answer for this question is option A, which is replication. Gene transfer is also a function of plasmid. Gene expression is also, and plasmid is antibiotic resistant too. So the correct answer for this question is replication. Replication is not a function of plasmid. The next question is, which of the following is not a benefit of genetically modified crops? Increased resistance to pests and diseases, improved nutritional value, reduced use of herbicides and pesticides. D. Increased tolerance to abiotic stresses. So option B is not the benefit of genetically modified crops, which is improved nutritional value. So other three options are very much correct, that is A, C and D. But which one is not correct is B. तो बायोटेक्नोलॉजी के कुछ क्वेश्चंस हम लोगों ने आज डिस्कस किए, और भी कमिंग वीडियोस में मैं और भी बायोटेक्नोलॉजी से रिलेटेड इंपॉर्टेंट एनसीईआरटी एमसीक्यूज अपलोड करूँगी. सो स्टे ट्यून्ड विद माय चैनल, एंड इफ़ यू फाइंड दिस वीडियो हेल्पफुल देन डू लाइक, शेयर एंड सब्सक्राइब माय चैनल फॉर मोर वीडियोस लाइक दिस. थैंक यू.